Hi, my name is Gabriel Rojas Bowe, and today I'll be talking to you about how prelimbic neurons can signal approach avoidance conflict. Firstly, we have to discuss what constitutes approach avoidance conflict. Take this image as an example. Here, we have two mice that are probably hungry. They know that to satisfy their hunger, they must go outside their hole and search for food. However, we also see that there is a cat waiting outside. Although the mice want to search for food, they also want to avoid an undesirable encounter with a predator. This image portrays an example of approach avoidance conflict. The question now is, how will the mice resolve this conflict? Will they wait until the cat goes away? Will they risk getting caught and make a run for it? Or will they decide to wait until the cat looks elsewhere and try to sneak behind it? The cognitive processes behind these different strategies to approach avoidance conflict are what interests us in the lab. Now, how do we study adaptive responses to approach avoidance conflict? Firstly, we have what we call the low conflict task. Here, hungry rats are placed in a cage with a lever that they can press for food. During the task, rats press the lever every now and then to receive a random sucrose pellet. Rats learn that a tone predicts a foot shock and that by mounting onto a nearby platform, they can avoid this foot shock. However, in order to avoid, the rat has to cease pressing. Here is a video of how this behavior looks. We can see that the rat quickly ceased pressing and mounted the platform in order to avoid the impending foot shock. However, this situation does not induce a high state of conflict. Even if the rat has to cease pressing for food, it can resume pressing once the tone ends and keep getting food, hence the term low conflict. In order to increase the level of conflict, we modified this task. Now, in the high conflict task, rats learn that a tone predicts a foot shock, but they also learn that food is only available when a light cue is on. What we do is that we then co-present the tone and light cues to the rat, forcing it to decide between mounting the platform or pressing the bar for food. What rats learn is that, although the tone is on at the same time as the light, the foot shock only comes at the end of the tone, which lasts 30 seconds. Therefore, what they do is that they press early during the co-presentation period and then mount the platform halfway through the tone before the foot shock comes. Here is a video showing the behavior. So, the prelimbic cortex, or PL, has been shown to mediate platform-mediated avoidance, as well as encode reward and reward-related behaviors, and it has also been found to be necessary for flexible decision-making. So, the question stood, does PL mediate approach avoidance conflict? We started pharmacologically inactivating PL during low conflict using the GABA A agonist mucimol. When we inactivated PL, we found that avoidance was delayed, as you can see in the graph below, where the animals infused with mucimol in orange and the saline controls in gray. However, when we did the same thing but during high conflict, we found the opposite. Avoidance was accelerated. These opposing findings suggested that PL was somehow detecting the high conflict scenario and switched from promoting avoidance to delaying it, suggesting that PL promotes the most advantageous strategy. In low conflict, this would be avoiding quickly. In high conflict, however, this would be avoiding later in order to accommodate pressing during the start of the tone. Given this, we were now interested in how PL might be detecting this conflict. So, this led us to our next question. Does PL signal conflict at the single cell level? To address this, we performed single unit recordings in animals trained in high conflict. What we first saw was that PL neurons can distinguish tone alone versus tone plus light stimuli. In this Venn diagram, I'm showing the proportion of neurons that were responsive to the tone only, the tone plus light only, and then in the middle, neurons that responded to both stimuli. Now, in the left top, I'm showing how the activity of neurons selected to be responsive to the tone alone look like under the tone plus light stimulus. And in the bottom, you can see how the peak response to both stimuli look in these cells. Now, to the top right, I'm showing the opposite. Neurons selected to be responsive to the tone plus light stimulus and how they look when presented with the tone alone, as well as the same peak response graph here in the bottom. Now, taken together, what these data suggest is that PL is able to distinguish the tone alone stimulus, which would correspond to low conflict, from the tone plus light stimulus, which would correspond to the high conflict. Now, what about the behavior of these animals? We first started looking at pressing. Now, there are two types of presses that can occur. Presses during light alone conditions, or no conflict, and presses during tone plus light conditions, high conflict. In the bottom graph, I'm showing how cells responded to both kinds of presses, yellow being light alone presses and blue being tone plus light presses. To the right, I'm showing a similar Venn diagram as before, but for pressing responses. As we can see, there are PL neurons that respond exclusively to one of the two kinds of presses, as well as neurons that respond to both. Taken together, these data suggest that PL neurons can distinguish presses during high conflict versus non-conflict conditions. Now, what about avoidance behavior? Well, although our analyses are still ongoing, we are seeing that PL neurons are distinguishing platform mounts that occur during high conflict versus platform mounts that occur during low conflict conditions, as you can see from the Venn diagram here on the right. So, conclusions. We first see that PL delays avoidance to allow early pressing, as suggested by our musimal data. We also see that PL distinguishes high conflict from low conflict cues, and that PL also signals pressing and avoidance behavior during high conflict differently than during low conflict. Now, future directions of this would be to record PL neurons throughout conflict training to see how they're learning to resolve this conflict. After that, we will want to identify targets of these PL neurons to map out this conflict circuitry. So, where are these neurons going? And then optogenetically manipulate these projections to establish causality in the conflict circuit. And so for acknowledgements, I'd like to thank the Quirk Lab, the now Bravo Lab, and of course, everybody in the Bravo Lab. And my funding source would be the MARC program at the UPR. And so, thank you. Now that we saw how rats detected and resolved this conflict, we want to see what brain region could be responsible for these neural processes.